Let me just start by saying thank you to Stacy and Robin for setting me up remotely. I couldn't make it out there in person, but I'm really glad to have the opportunity to um, get this across to the audience at 416. So I'm going to narrow in on a very specific case study, and I think that the previous two presentations were a great segue. It sets the stage for what I'm going to talk about. So I am earlier career stage. I'm a project scientist right now, starting as an assistant professor at UC Riverside as of July. So as I'm embarking on the tenure track, right away I'm already starting to think about my alternate products. So things which are traditionally not scientific products such as manuscripts, but other activities that I'm involved in. So today I wanted to talk specifically about software development as a non-traditional research activity. So I'm a marine biologist, but I work with high throughput genomic data. And because the data sets that we generate are so large and so complex and fairly new in terms of how long they've been around in science, we're really dealing with the fact that we have to do software development and we have to develop new pipelines as we're trying to analyze these genomic data sets in order to get any useful biology out of them. So I've been sort of pulled into the world of software development, kind of in a meandering path. But I think a lot about how this activity of doing software development is really viewed by my peers. So in terms of tracking my accomplishments, thinking about tenure portfolios, software development still is not really rewarded or valued as much as hypothesis-driven research. And this is particularly true in biology, where we're used to designing studies and looking at organisms. Genomics has kind of changed the game a little bit because now we're actually doing a lot more things on computers and a little bit separated from the organisms themselves. So there's a lag time in citations for new software products. A lot of times the type of software we're generating is scripts that we write on the fly to analyze a specific data set. We might put that on GitHub, we might publish it in supplemental material for the paper, or we might go full on and sort of package that into something more formalized in terms of an algorithm that we might also write a software paper for. But the problem that I'm facing, and I think many other people are too, is that there's a lot of software out there. We're all doing a lot of the software development, but it's not a very traditional research activity. So we're struggling with how to track dissemination of these types of products and usage of these products. And so they kind of have to be measured in other ways. And I think for me, as I'm trying to gather statistics for various end goals, I think a lot about what types of altmetrics are useful and are going to give me information that I can use moving forward. So specifically, I'm going to be talking about a software package that I was leading development of through the Alfred P. Sloan Foundation. So I was am principal investigator on a grant to develop a research-driven data visualization framework for high-throughput environmental sequence data. So this is a novel software framework to try and bring in more visual analytics into the analysis of genomic data sets. And really, this grant was to develop a prototype software framework, which hopefully will be adopted by the community and used by other researchers. The specifics. So I won't talk about the specifics of the framework too much, other than the fact that what we're really trying to do is build a Kayakcom for massive genomic data sets. So traditionally, when we're dealing with sequencing data, genomics data, we deal with a lot of large text files on the command line, and that's just really time consuming, not the most appropriate way to get biology out of these data sets. So if you had something like Kayakcom, where you could upload a data set and have slider bars on the left-hand side, you could have things that you could tweak, boxes you could check, uncheck, and then have a central space where the data was updated in real time, and you had some sort of visual information that you could use with your brain to process. That's what we were trying to do with the software framework. So what actually came out of that is a software called Finch. So it's online at finch.org, and this is what the visualization framework actually looks like. It's a browser-based tool. It uses JavaScript and D3 to visualize these genomic data sets, and this is just an overview screenshot of the different type of visualizations that are available. And the preprint is currently on bioRxiv right now. So there's different uses for this tool. One use is for research. So researchers who want to very quickly look at their data and get it into a visual context and make decisions about further sequencing or about papers that might come out. But another use of this framework is for education and outreach. So I've been working with citizen science projects to visualize genomic data sets that are actually sent in by the public. So this is the case where people were swabbing dust in their homes and submitting it to North Carolina State University. So these are microbial data sets being generated. And one of the problems is that citizen science don't have a research background. So, the challenge is really to find novel ways in which they can interact with these large data sets that they're helping to generate. So there's different audiences for this tool, and what we've struggled with in terms of Altmetrics is really capturing the usage of our tool, our framework, across these different types of audiences. So I'm just going to present a little bit of data from here. I'm not sure if I have answers, but I can just give you some insights on what we're actually trying to do and what sort of metrics we're trying to track in terms of grant reporting and renewal funding and those type of reporting requirements. So as I said, one of the main questions I'm interested in is we put the software out there, we developed it, it's on GitHub, it's on a website. So we really want to know who is using this Finch framework. And this is prototype software published in October 2014, so only about a year and a half ago. It's still fairly new. We're still spreading the word about it, actively trying to get back in developing and adding features after the prototype was stabilized and published out there. So this is, metrics that I'm tracking over a year and a half since the inception of the software, a very short time frame. So one of the first things we did is, because it's a web-based framework and because it's a website that people are going to, we can track website analytics, which is not something you typically think of as a research scientist that you should be keeping track of, but something we found pretty useful in terms of just figuring out how many people are accessing the site on a day-to-day or month-to-month basis. So in terms of website analytics, we can track geographic distributions. Most of our users right now are coming from the US, but we do have some uptake in the US, Canada, and then lower amounts of uptake from other countries around the world. In terms of actual users that are coming to the site, so we can look at peaks and in usage in terms of how many people are accessing the site across days and months, like I said. And really what we're interested, I think, in are some of these more cumulative statistics in terms of the number of page views. So this was a 2-month period in 2014 that I was tracking. And so we recorded over 3,000 page views, 50-50 split in new visitors versus returning visitors, which is good because that means that new people are coming to the site fairly regularly, and session duration. So this tells us a little bit that people are actually using the tool. They're not just clicking a link in Twitter and then closing the browser window, but average session duration is above 7 minutes. So hopefully that means they're actually staying there and uploading some data or playing around with our test data to try and explore the software a little bit more, which is all pretty good news for me in terms of disseminating the software and getting people to use it. Other things which I've noticed, and I'm on the fence about whether or not this is a good thing or a bad thing, is the impact of in-person regarding the software framework. So this is just a screenshot of website analytics over a couple months in 2014, and this coincided with some conference talks that I was giving. So I went to the Society for Molecular Biology and Evolution conference, and then I gave tutorials about the software at a undergraduate workshop and a microbial ecology workshop. And so this kind of reinforces the idea that even though this is an online tool and it's digital for all intents and purposes, the act of me personally, physically going to conferences actually resulted in a lot more page views and a lot more users actually accessing the site. So that sort of makes me think that this is really important. An important complement of doing software development is actually getting out there and almost being like a salesman at these type of events and going out and selling it for different types of purposes. The other thing, which is great, is that we use GitHub. So this is where the open science aspect comes in. So the code base is actually on GitHub. It's free to reuse or adopt. And GitHub is a code repository that provides its own set of statistics. So these are website analytics essentially for the code itself. So I've been tracking these as well. The address for the software is on the bottom if you're interested. But really, in terms of the code base itself, we can look at end users, so people that are favoriting the repository essentially. So that's kind of bookmarking the repository in case they want to use it or look at the documentation. We also have what's called forks. So forks is if you want to duplicate the code base and start making changes yourself, that shows up here as well. So this is not only the end user, so the biologist with no computational experience, but it's also the software developers who are out there in the community that might be looking at our code and modifying it and using it, which is great because we're trying to build up a community around this open source framework, and we're not, we don't have licensing, we're not charging people to use the software. So getting more people involved in the development is always a good thing as far as we're concerned. Here are some more stats from GitHub. So like I said, GitHub can sort of give us website analytics from a different perspective for the code base itself. So here we can have another view of referrals. So how are they getting to our site? We see that they're coming from GitHub itself, but also Google searches, our browser, public implementation of the code. They come from there, as well as Authoria, and some other institutes like the USDA. We can also see that people are looking at the wiki, so they're looking through the documentation, they're trying to use the software, and we can see the most popular pages are sort of the start guide, the homepage for the software, as well as the quick start, which is just the easiest way to get started, kind of a cheat sheet to what you need, which is, it's nice to see these numbers. I'm not quite sure how they're useful, but in terms of collecting them, it's great that it's possible to actually get these statistics automatically by just putting the code online in an open source repository. So we can also look at issues. So issues are kind of like a help forum for users or developers. If there's something they have a question about, they can post them in this group forum. So we can see how many issues have been opened, over a period of time, cumulatively. We can see how many issues we've engaged with, how many we've closed or answered, and just generally track the activity in terms of questions and answers between the developers, so my end, the people who are developing the software, and the people that we're hoping are using the software. There's other things too. So in terms of the actual where our project is housed, we're tracking that, but we're also trying to use different social media metrics to see how much this software is being disseminated. So this is the case where I'm trying to track documents that are related to this software project and seeing how many people are actually reading them or sharing them. And so I actually posted the grant proposal itself on Figshare. Before the software was even online or had been developed, I had sort of put this on Figshare to announce to the community that it was in development. And so I have been tracking this on impact stories, so getting Figshare views, downloads, views on Twitter, and impact story itself. And we have badges for these. It's showing up as highly viewed, highly discussed, and coming into the 99th percentile on impact story for similar types of products, which is good, hopefully. Another thing that I've been tracking as well is the manuscript preprint on bioRxiv. So this was posted in 2014 while we're trying to get the software paper peer-reviewed and edited. So this is, again, some stats from Impact Story and Altmetrics, just showing how we compare to other papers that are out there. So we have an Altmetrics score of 15 right now. Our preprint is in the top 25%. We're mentioned by 24 people on Twitter, and we have a timeline of when that activity happened. So really it was as soon as our preprint went on bioRxiv, we had a flurry of tweets, which has since died down, which seems similar to some of the statistics that were quoted earlier. And the demographic breakdown is sort of 83% scientists versus members of the public. So these types of numbers are things we're gathering, as I said, reporting in grants. I'm keeping track of them. I like to look at how my in-person interactions, as I said, the conference talks or workshop demonstrations, how those actually translate to these type of metrics. Why Altmetrics? So why am I doing this? It's very time consuming, but I think hopefully it should be important in the long run in terms of planning my own strategy. So figuring out what works, what doesn't. And like I said, I think in-person talks that I'm giving are fairly effective in driving traffic to the website. But really what my main goal is to understand how effectively that I, as principal investigator of this grant, is really disseminating my research products. And as I go forward, as I start and have a lab group, I really want to be able to track my group's research products, because I am probably going to have a lot of these non-traditional outputs, such as software. So, you know, tracking things like who is using our tools, how are they discovering the software, and are we really meeting the end user needs? So if we want a community to build around this tool, we have to engage with the community, and we have to make sure that we're not developing this in isolation and being completely fooling ourselves about what people want and need in this type of software for genomics. For an open source software, you know, we're really keen to build a community around this. We're not getting royalties. We're not generating revenue streams through software licenses. So in an ideal world, we would really be able to encourage and foster the growth of a user community, but also a community of other software developers who can help us sustain and grow this type of data visualization framework in the long run. But really what I'm struggling with now, and I think that this is common for all of us, is figuring out what these numbers really mean. So it's great that we can track them. And I think for me, I think it is important that I do track them, because I can have graphs and different statistics that I can show to people if they ask. But in terms of, you know, in the long term, generating citations, generating people who are going to be using this tool and putting it in their research paper, I'm not convinced that these numbers are going to tell me anything or going to be able to predict anything. So with that, I'll just say thank you to my collaborators and again to Stacy and Robin for inviting me. Thank you.